joining us for Amplia Therapeutics shareholder webinar this morning. Presenting on the call today, we have Dr. Chris Burns, Amplia's CEO, and Dr. Rhiannon Jones, who is Amplia's COO, who will be doing a short presentation, and we will follow that with a Q&A. If you would like to ask a question, please do so using the Q&A button at the bottom of your screen. Otherwise, if we don't get to all of your questions today, we do apologise, and we will address them in the future. So for now, I'll just hand over to you, Chris. Thank you.
Thanks, Hannah, and thanks to everybody who's joined the call today. We're really thankful that there's been so much interest in our recent data, and thankful for you to take your time to hear us speak a little bit further about that. And I'll hand over to Rhiannon as I pull up some slides. Maybe Rhiannon should be driving this. Apologies. And why can't I see that? Okay, that's a little bit odd. So, okay, apologies while we sort this out. Do you know how to do that? I'm not seeing.
Let's try sharing the entire screen, which is unfortunate, but we might just have to do that.
Sorry about that. Thank you. Yeah, so I'll hand over to Rhiannon to lead us into the presentation.
Thanks, Chris. There are some forward-looking statements in this presentation. I'm going to start by taking you through pancreatic cancer, which most of you are likely already aware of. It's a devastating disease and the third most common cause of cancer death. It's often diagnosed late. Indeed, over 50% of people diagnosed with pancreatic cancer are diagnosed at an advanced stage. And at this point, there is only a 3% survival rate, 5-year survival rate. It's a large market of $3.8 billion US, and it's predicted to grow to $14.4 billion by 2034. One of the reasons that pancreatic cancer is so hard to treat is due to this dense fibrotic matrix that surrounds the tumour tissue. This makes it difficult for chemotherapy and other agents to penetrate into the tumour. Nalmefrotinib helps to reduce this fibrotic barrier to allow the chemotherapy and these other agents to penetrate. And then nalmefrotinib and these agents can act on the cancer cells themselves to kill them. So like this springboard here and these divers, the springboard helps the divers to leap higher, and nalmefrotinib helps these chemotherapies and other agents to work better. Nalmefrotinib has these 4 main advantages that we've seen in our clinical trials. We see an improvement in efficacy. We see good tolerability and safety. We see that there is a longer duration on study, and nalmefrotinib has the convenience of oral daily dosing by a capsule. Our ACCENT trial is our first clinical study in pancreatic cancer. It's testing the combination of nalmefrotinib with the chemotherapy, gemcitabine and Abraxane, in metastatic pancreatic cancer. The objective of this study is to look at the safety and tolerability of this combination, but also to look at the efficacy via the overall response rate, which is where patients are scanned by CT every 2 months and the tumour size is assessed. The study was split, or has been split, into 2 parts. In part A, we have looked at increasing doses of nalmefrotinib in order to choose a dose for the phase 2 of the part B of the study. And in part B, it's a single-arm, non-blinded study in a Simon 2-stage design. In part B, there were 55 patients, and in part A, 15 patients. And in total, 64 patients have been dosed at this 400 milligram dose. We've recently sent our data for independent review. This is where the scans are sent to an independent contract organisation where two experts look at these scans and assess them separate to each other, and also without knowing the initial assessment and without knowing any of the clinical information from the patients. This is the preferred method of review by the regulators in open label single arm studies. Previously, our data has been read at the site. This was needed so that we could have a real-time assessment of how patients are going and indeed how our study is progressing. But it's important to note that RESIST 1.1 analysis, which is what is used to read the overall response rate, is standard in clinical trials, but not in clinical practice. We have always intended to send our data for independent review at the completion of the trial, but we had this brought forward so that we could progress our development into further studies. Shall I hand over to you, Chris, to talk about this further?
Thank you, Rhiannon. Yeah, so the benefit of the independent review, I think, as Rhiannon has noted, is that it's a standardised review, blinded and preferred by regulators. And what we reported yesterday to the market is that we've seen an additional 4 complete responses. So we had previously announced 1 complete response in the Part B of the study and 1 pathological complete response. They were announced midway through 2025. And what we've seen now from the independent review is there's an additional 3 complete responses in the Part B, so 4 total complete responses in that 55 patient cohort, and indeed a complete response in Part A. So if we remember, Rhiannon noted that there were 9 patients in Part A that were treated with 400 milligram dose of nalmeflotinib on a daily basis prior to chemotherapy. We've now got 1 complete response and 2 partial responses from that cohort. And additionally, in Part B, so the 55 patients, we've now got 4 complete responses and 16 partial responses. So if we put that into a table of response rate and compare it with the MPACT trial, the MPACT trial led to the approval of gemcitabine Abraxane as standard of care for chemotherapy, and it is widely used around the world as a treatment for pancreatic cancer that's metastasized. And we think it's a very appropriate benchmark for our study, because our study indeed was designed including many of the criteria that were present in the MPACT study. So in terms of inclusion and exclusion criteria of patients and other details around the protocol. So as we look at that comparison, we see the fact that we've seen 4 complete responses in Part B alone. So just looking at the 55 patient cohort there, that's a 7% complete response rate. That compares to a less than 1% response rate, complete response rate in the MPACT study. So that was a study of 431 patients. They saw 1 complete response. So we've seen 4 complete responses in 55 patients, or indeed 5 out of 64 patients. In terms of partial response, we're seeing a 29% response rate. In terms of stable disease, 33% response rate. You can see across the board there that we have a better response rate compared to the MPACT study, such that the overall response rate is 36%. So that's the complete response versus partial response. What I should note, these data also compare very favourably to other clinical studies where gemcitabine Abraxane has been used in the same way that we've used it in the ACCENT trial, particularly in terms of complete response and partial response. So again, we compare very favourably and are superior to what has been reported in the past. Next slide. The other thing that was important to release to the market was this measure of overall survival. So this is looking at how patients are doing overall in terms of survival. So even once they've left our study, how do they survive post treatment on our study, and indeed moving into second studies and beyond. And what we see here is that in the MPACT study, the overall survival was 8.5 months, and more recently in the NAPOLI-3 study that looked at gemcitabine Abraxane in a control arm, they saw a overall survival of 9.2 months. So essentially around 9 months, if you like. We're seeing an overall survival of 11.1 months. So that's a full 2 months improvement over what's been reported previously. That again is very meaningful for patients, but importantly, it's also meaningful for regulators. The last drug that was approved in pancreatic cancer in the US was the drug regimen called Nalirofox. That was approved in the NAPOLI-3 study, and that was approved on an 11.1 month overall survival. So we have identical, strangely enough, identical overall response rate to Nalirofox. But with a different chemotherapy regime. Ours, of course, is in combination with gemcitabine Abraxane. And importantly, on the next slide, we can have a look at the adverse event profile that we see in our trial, what was been recorded for the MPACT study, and what indeed was reported for Nalirofox in its approval study. So that's the study that led to the 11.1 month overall survival and approval of Nalirofox. So what you see there on the blue column is that our profile is very similar to what you would expect from gemcitabine Abraxane alone, and we've reported this data previously, showing that nalmefotinib is not exacerbating any of the toxicities or tolerability issues that we see with chemotherapy. Indeed, in some cases like peripheral neuropathy and anemia and so on, that it is very, very favourable in terms of what you'd expect with gemcitabine Abraxane. So we see a really very, very good response in terms of tolerability in the ACCENT trial. If we look at the far right-hand column, this is the data from the NAPOLI-3 study of Nalirofox. And what you see here, and we've grayed out those sections where it's particularly noteworthy, is things like diarrhea and nausea and vomiting are quite significant in FOLFIRINOX treatment and indeed Nalirofox treatment. Nalirofox has a slightly better tolerability profile to FOLFIRINOX, which is the old standard, but it does have these quite significant tolerability issues around GI toxicity, and we don't see those in our study. So I think the take-home message here is that we have the same median overall survival as Nalirofox, but we have a better toxicity profile, and importantly, we don't have a different toxicity profile to what we see with gemcitabine and Abraxane alone. So to summarise that in sort of really simplistic numbers, we've had a really good response rate. We've got 5 patients that have had a complete response, so complete disappearance of tumour and metastasis for 2 months or more. We've got an improved overall survival of 11.1 months versus 8.5 months, so a 1.3 times improvement over what you'd expect for chemotherapy alone. And lastly, we have a very good safety and tolerability profile, where the additional safety burden to patients and the additional effects of nalmefotinib are minimal. So if we think moving forward, what does this data set us up for? Well, we've spoken publicly about our interest to expand into some new therapeutic indications, as well as moving forward with a registration-enabling study in pancreatic cancer. So what we have shown here is that there's a number of cancers where FAC involvement has been clearly identified from scientific and genetic studies: pancreatic, colorectal, lung, ovarian cancer. And so there's a lot of interest, obviously, in pancreatic cancer, which is what we've been focusing on, but there's interest in other cancers as well. And then if we look at combinations that a FAC inhibitor, namafotinib, could be used in, there's clinical data of our own, and then there's preclinical data and indeed published studies showing that FAC inhibitors can combine with targeted agents, so for more personalized medicine, radiation therapy, biologicals like antibody drug conjugates, and indeed even cell therapies. So we see a big opportunity to combine our FAC inhibitor with a range of other different treatment modalities in a range of other cancers. We've spoken before about ovarian cancer, and we hope to report further on that in due course. So if I finish up just mentioning that we do have a conference presentation lined up at the American Association of Cancer Research in April, so in about a month's time. We'll be presenting further analysis from the independent read data at that study. We hope to have that deeper analysis completed at that time. We really only received this data recently. And in terms of timelines, you can see here we are working with a local charity about initiating an ovarian cancer trial. We'll announce to the market when and if that gets off the ground. That should start sometime this year. We expect to continue our work in the Amplicity study. So this is our second study in combination of namafotinib and FOLFIRINOX. And we're also talking to regional partners in the Asia Pacific region about licensing or partnering deals, which we anticipate the conclusion of some of those discussions by the end of this year. Then next year, we're doing a lot of work now, but we're building up to initiating a phase 2b/3 study that will be registration enabling of namafotinib in pancreatic cancer. The whole field of pancreatic cancer is changing significantly, so we've got a lot of opportunity there, and we're working with the regulators and our internal teams to submit an application to the FDA for final approval. And then looking further ahead, the Amplicity trial, we expect to see that complete in early 2028. And as we continue to develop data, we plan for a licensing or partnering deal for the rest of the world for namafotinib. So hopefully that gives a flavour for where we are with our current study and the plans going forward. So we'll stop there. There's Rhiannon and my email addresses, so please feel free to reach out to us if there are any additional questions. I know we've got a number on the from the webinar, and we'll try and address those now, but if we don't, please feel free to reach out to us and we'll come back to you on those. So we're going to stop sharing and pull up the questions. So Hannah, are you happy for me to just walk through the questions that we have?
Yep, there are a few that we got in advance that I may just read out for you. The first one being, it's great to see that we've had 5 patients with a complete response. What makes those patients so special that they responded better to the treatment?
So thanks for reading that out, Hannah. So yeah, we've got a few questions along this this line of thinking. Why are those patients, and indeed why do we see a whole cohort of patients respond very well, quickly, as Rhiannon said, with a good duration of response and a deeper response? This is an area of considerable focus within the company now, both with preclinical studies and indeed looking at clinical samples. We don't have any specific data to elaborate on at this point in time, but we are actively reviewing the genetic profile of patients' tumours, where we can, an assessment of the fibrotic nature of those tumours, to see if there's an indication in one of those readouts about why certain patients have responded better than others. So I think that's a really important area in biomarker analysis that we're working on. Next one.
Next one. Given that narmofotinib achieved the same 11.1 month median overall survival as nalirifox, but the complete response rate for that was negligible at 0.3%, and it also had severe toxicity. How prominently do you expect this comparative therapeutic feature in your discussions regarding partnerships with global pharmaceutical companies?
Yeah, look, that's a great question. Obviously, the end game here is for us to partner with larger players to take the product to the market. We are constantly having discussions with pharma and biotech who are working in the GI cancer space. We are continuing those discussions. We have reached out to some of those parties with this newer data, and indeed, I think this will enhance the ability to move those discussions forward. And I think it's also noteworthy in terms of dealing with the regulators, and I'll just pick up sort of a sidebar to that comment, is that we can reassess the opportunity to look for some accelerated programs like breakthrough designation, given the fact that the level of complete responses we've seen to date is unprecedented.
Yep. Okay. What is the average survival of the complete response patients since treatment, and do these patients lose all tumor markers as well as being CT scan negative?
Yeah. So look, that's part of the ongoing analysis that we're doing. So we can't speak to individual patients and that kind of analysis at this point in time, but we do hope to have that analysis completed by the time of the AACR presentation.
All right. Number 4, what was the key driver between the difference in complete response rate between the investigator radiologist assessment versus the central lab?
You're going to take that one again?
Sorry, what was the
driver between the difference of the original data and then the revised data from the independent lab?
As I mentioned in the webinar, these independent experts, they review these daily. And so they are experts at reviewing these scans. They do it independently. They are unbiased because they don't see any of the additional clinical data. Whereas at the sites, RESIST really isn't used for clinical practice. And so it is outside of standard clinical practice at the investigator level.
It's worth noting we always anticipated there would be a difference between the investigator-read and independent-read data. That's well known in the field, in the scientific literature, and we anticipated there'd be a difference. Obviously, we were very pleasantly surprised to see that the independent read was identifying some additional complete responses. So that's that's just the nature of clinical practice and clinical studies. But I think, as Rhiannon said, that the intention was always to have our data read independently at the end of the study. So we're now undertaking that process.
All right, thanks very much. Next question. These are the ones that are coming in live. I'm just reading from the screen. It says, given that 4 patients are still on the trial, can we expect the median overall survivability to increase further?
So the median, the way the statistics work, the longer that some people stay on study is not really going to change the median overall survival substantially. It's obviously fantastic news for those patients. We've got one patient who next month would have been on study for 2 years, and all other patients who remain on study are actually doing incredibly well. So we would expect a small change in overall survival, but not a significant change.
What would the trial size look like for a phase 2b/3?
So that's a great question, and one that we're discussing with our regulatory advisors and indeed with some clinical trial groups at the moment. The standard sort of size for these studies, as I mentioned, the Impact study had 430 patients. I think these days you can pare that back to somewhere between 200 and 300 patients per arm under a normal clinical trial protocol. But what's happening in this space, we're getting a lot of development in things like biomarkers that predict for response. We're coming up with new trial designs that allow a more efficient development of clinical studies. And indeed, you will be aware, many of the listeners will be aware of changes at the FDA as well to help expedite the development of drugs in oncology. So at the moment, it could be 200 to 300 patients per arm, which is obviously a very large clinical study because we'll have an active arm and a control arm. But we are looking at ways and working and discussing opportunities with some other groups where we could bring that number down quite significantly.
All right. We are running out of time, so I'll just do a few more. Why do you think namafotinib has lower GI issues?
Well, its mechanism of action is very different to the chemotherapy. Chemotherapy, the way it works, it targets any rapidly dividing cell, and that's often cells you have in your gut. So chemotherapy is often associated with gastrointestinal toxicity. Our drug targets very selectively a protein called FAK. We don't see a lot of GI toxicity, presumably because FAK activity is not particularly significant in the gut under normal conditions. So I do want to acknowledge that the drug was discovered here in Australia by a really talented group of scientists at the Cancer Therapeutics Cooperative Research Centre. And I think a lot of the clinical data that we're seeing, the promising safety and tolerability, really goes back to the excellent work that team did in optimizing this particular molecule. And it's why we were so keen to license it, because we knew it was a very quality drug that had a lot of excellent attributes. So I think a lot of this comes out from the quality of the science that went into the drug itself.
Thanks, Chris. Okay, where does manufacturing take place, and is it all done under GMP standards?
Joanne, do you want to say that?
Yeah, all of our manufacturing is done at a GMP facility and to GMP standards. That's what's required for this level of development. We are ensuring that our manufacturing processes grow and evolve as our clinical development grows and expands, and so that we are always manufacturing to the appropriate standard for our level of development. Yeah.
Okay, so last question. It just says, on dosing, with the ACCENT phase 2b/3 trial, will you use 400 milligrams, or would you possibly increase the dose to 600 milligrams?
Yeah, it's a very excellent question. So at this point in time, we've used 400 milligrams based on the circulating drug levels that we've used 400 milligrams, sorry, based on the circulating drug levels and the safety and tolerability we saw in the 1B study in ACCENT. We do intend to actually test that higher dose of 600 milligrams once a day. That's part of our optimization. That'll be part of the phase 2B study that we've been talking about, the 2B/3. At this point in time, we don't know whether that will be as well tolerated as 400 milligrams, but we would like to push that dose and see if we can get further improvements in response. And the other point to note is in the ACCENT study, namafotinib is given intermittently. So patients take the drug for 4 days prior to chemotherapy, then they have a couple of days off. We actually want to go to a daily dosing regimen, which is what we're using in the Amplicity study. We think the benefit of this is obviously easier for patients, but it's also an opportunity, I think, to keep the level of drug in the circulation higher, and we believe we'll get a better therapeutic outcome based on our modelling and studies to date. So short answer is yes, we will look at the 600 milligram dose, but we think the 400 milligram dose is a very good dose nonetheless.
All right, thank you very much, Chris. We will wrap it up there, given we've reached time. To anybody, if we didn't get to answer your question, I'm really sorry. You have the contact details, so please send them through. Before we close, do you have any last remarks, Chris or Rhiannon?
No, just well, nothing specific other than to thank everybody for their time today. We're really excited with the data we've been generating. It's great to have the support of our shareholders and other stakeholders. I want to acknowledge, obviously, all the clinical trial participants and the clinical trial sites for their involvement in the study, and we look forward to presenting further data at the AACR meeting in next month and sharing that presentation with you publicly when that's available.
All right, thank you very much both, and thank you everybody for joining. We look forward to updating you with new information in due course.
Thank you very much.
Thank you.
Bye-bye.